Most people think they know what the DNA of African Americans would show. Broad West African ancestry, filtered through slavery, end of story. That is not what the science found. The genetic data is more specific and more disturbing, and in several places more complicated than the general history suggests. And some of what it reveals was not supposed to be recoverable at all. This is what the research actually shows. Before we dive in, comment where you are watching from and click subscribe. Most immigration records tell you something. A ship manifest from 1890 lists a name, a village, and a country of origin. Ellis Island processed millions of arrivals and recorded where they came from with enough specificity that descendants today can trace a great-grandparent to a specific town in Sicily or to a county in County Cork. That infrastructure of origin, imperfect as it was, existed because the people arriving were considered people. The records kept for enslaved Africans were different in kind, not just in quality. Slave manifests recorded cargo. They noted age, sex, physical condition, and monetary value. What they almost never recorded was ethnic origin, regional identity, language group, or the name a person had before being given a new one. The word used was simply African. An entire continent was collapsed into a single administrative category, not out of ignorance because the traders knew exactly where their ships had been, but out of deliberate indifference to information that would have made the people harder to think of as property. This was not a bureaucratic oversight, it was a system. Names were changed on arrival, languages were suppressed, sometimes violently. Enslaved people from the same ethnic group were sometimes deliberately separated to prevent communication and collective resistance. The knowledge of where a person came from was not just unrecorded. In many cases, it was actively destroyed. For generations of African American families, this erasure was the wall at the end of every genealogical search. Census records, church documents, and Freedmen's Bureau files could sometimes take a family back to emancipation in 1865 and almost never further. The names before that point were often the names of enslavers. The places before that point were simply gone. Historians understood the scale of what had been lost. What they did not know and could not have known until recently was whether any of it was recoverable. The ethnic origins of millions of people were suppressed for centuries by a deliberate policy encoded in no surviving document. The question was whether that information had survived somewhere the system could not reach. It had. It was in the DNA. The earliest genetic studies of African Americans were not designed to recover ethnic origins. They were designed to answer a different question entirely. In the late 1980s, and the early 1990s, population geneticists were mapping mitochondrial DNA diversity across human populations worldwide, trying to understand the deep architecture of human migration out of Africa. African Americans entered those studies as one data set among many. No one was specifically looking for the slave trade in the DNA. What they found there anyway was the first indication that something specific and recoverable was encoded in those sequences. The expectation going in was straightforward. African Americans had been transported primarily from West and Central Africa. Researchers expected a broad West African signal, reasonably uniform across the sample population. That is roughly what appeared, but the uniformity did not hold up at closer resolution. When researchers began disaggregating the data by region, comparing African American populations from different parts of the American South, the haplogroup frequencies did not match. Coastal South Carolina and Georgia populations looked genetically distinct from populations in the Mississippi Delta. Populations in Louisiana carried frequency patterns that differed from both. These were not dramatic differences. They were statistical shifts in the proportions of specific maternal lineages. But in population genetics, statistical shifts in haplogroup frequencies are not noise. They are signal. They record history. The question was whose history and from where? Mitochondrial DNA can tell you which broad maternal lineage a person carries. Mapping that lineage back to a specific African population requires a reference database, a catalog of which haplogroups appear at what frequencies in which African populations. In the early 1990s, that database barely existed. African genetic diversity is the deepest on Earth, the product of the longest continuous human habitation of any region on the planet, and it was, and in many respects remains, the most poorly documented in the genomic literature, so the signal was there. The map to read it was incomplete. Researchers could see that something geographically specific was encoded in the data. They could not yet say precisely what. That problem would take another decade and significantly more ambitious research to begin resolving. And when it did, the results did not simplify the picture. They made it considerably more complicated. In 1999, a team of historians and economists began assembling what would become the most comprehensive database of the transatlantic slave trade ever constructed. The Voyages Project eventually cataloged records from over 36,000 individual slave ship voyages. It recorded ports of embarkation, ports of arrival, estimated numbers of people transported, mortality rates during the crossing, and the names of the ships and their captains. It was, in the most precise sense available, a map of the trade. Geneticists noticed it immediately. If mitochondrial DNA data showed that certain African American populations carried haplogroup frequencies consistent with specific regions of West and Central Africa, and a shipping database showed which African ports supplied which American destinations, the two data sets should align. When they did align, it was not coincidence. It was the slave trade itself, legible in the DNA of living people. The alignments that emerged were not vague. Populations in the coastal Southeast, particularly South Carolina and Georgia, showed elevated frequencies of haplogroups associated with Sierra Leone, Guinea-Bissau, and the broader Rice Coast. The historical record showed that Carolina planters had specifically and repeatedly requested enslaved people from that region because rice cultivation was the economic foundation of the Carolina colony, and the Rice Coast populations had cultivated rice for generations. The demand was documented, the supply route was documented, and now the genetic signature of that specific human traffic was showing up in the DNA of people living in the same geographic region their ancestors had been delivered to centuries earlier. The Louisiana and Gulf Coast signal was different. Those populations carried elevated frequencies consistent with Central African, specifically Congolese and Angolan origins. The Voyages database confirmed that a significant proportion of enslaved people transported to French Louisiana came through Central African ports. The genetic map and the shipping records were converging from two completely independent directions. But convergence is not the same as proof. Haplogroup frequency matching works at the population level. It identifies probable source regions. It cannot name an ethnic group with certainty, and it cannot account for the genetic diversity that was lost before the data could be collected. There is one population in the United States where the genetic signal is not statistical and approximate. It is overwhelming and specific. The Gullah Geechee people of coastal South Carolina, Georgia, and the Sea Islands have lived in geographic semi-isolation for over 3 centuries. Their language is a Creole, built on an English base, but threaded with vocabulary, grammatical structures, and tonal patterns that linguists traced decades ago to Sierra Leone, specifically to the Mende and Temne-speaking peoples of the Sierra Leone River estuary. That linguistic connection was documented long before anyone ran a DNA study. It was sitting in the language itself, waiting to be read. When geneticists sampled Gullah Geechee communities, the Sierra Leonean signal was not subtle. The frequency of haplogroups associated with Mende and Temne ancestral populations was significantly higher in Gullah Geechee samples than in any other African American population studied. This was not a broad West African match. It was a regionally and ethnically specific match, the kind that population geneticists rarely get to make with this level of precision. The third line of evidence was economic. The Carolina rice economy of the 17th and 18th centuries was not self-sustaining on European agricultural knowledge. Rice cultivation in tidal wetlands requires specific techniques that European settlers did not possess. Carolina planters knew this. The correspondence is documented. They specified in their orders to slave traders that they wanted people from the Rice Coast, the stretch of West African coastline running through present-day Sierra Leone and Guinea, because those populations had cultivated wetland rice for generations, and the planters understood that the expertise came with the people. 3 lines of evidence. Linguistics identified the Sierra Leone connection from the language. Genetics confirmed it from the DNA. Economic history documented the deliberate mechanism that produced it. None of these fields was talking to the others when they reached their conclusions. They converged independently. This is the cleanest case in the entire data set. A specific people, a specific origin, a specific economic logic that drove the connection, and 3 independent disciplines arriving at the same coordinates. The Gullah Geechee story is not typical of what the science can recover, but it shows what becomes visible when the conditions are right. Mitochondrial DNA travels through mothers. That is the only line it follows. A woman passes her mitochondrial DNA to all of her children, but only her daughters pass it on. The paternal line, the father's father's father, runs through the Y-chromosome, and it moves exclusively through men. When researchers turned from mitochondrial studies to Y-chromosome analysis in African American men, they were not expecting symmetry. They did not get it. The Y-chromosome haplogroup data showed something that the mitochondrial studies had not. A substantial proportion of African American men carry Y-chromosome haplogroups that were not West African in origin. They were European, depending on the study and the population sampled. The frequencies ranged from roughly 25 to 35%. In some regional samples, the figure was higher. These were not post-emancipation lineages. The molecular clock estimates and the specific haplogroup variants pointed consistently to the antebellum period. The timing placed those lineages inside slavery. This is the genetic record of what the historical literature has documented but rarely quantified. Sexual violence under slavery was systematic. Enslaved women had no legal standing to refuse. Enslavers and overseers operated without accountability. The children born from that violence were enslaved at birth, their paternal ancestry legally irrelevant and socially suppressed. What the Y-chromosome data shows is that this was not incidental. It was demographically significant. It left a measurable imprint on the paternal lineages of millions of living people. The asymmetry between the maternal and paternal data is itself a historical document. African maternal lineages survived in high proportions because enslaved women's reproductive lives, however violently controlled, transmitted their mitochondrial DNA to their children regardless of the father's identity. The paternal African lineage was far more disrupted, not because African men were absent, but because the conditions of slavery suppressed the transmission of their Y chromosomes through generations of coerced reproduction. For many African American men, this is not an abstraction. Their direct patrilineal line, the lineage that Y chromosome testing traces, does not lead to West Africa. It leads to Europe. The DNA is not making a claim about identity. It is documenting a specific and measurable historical violence. Those are not the same thing, but they are not easy to separate either. Mitochondrial DNA and Y chromosome studies each trace a single line of descent. Together they cover 2 threads out of the thousands that make up a person's actual ancestry. The rest lives in the autosomal DNA. When researchers began applying genome-wide analysis to large African American samples, they were not adding a third data point to the picture. They were finally looking at the whole picture. The findings from large-scale autosomal studies, including analyses drawing on data from hundreds of thousands of African Americans conducted in 2010, and again with greater resolution in 2015, produced a continental ancestry breakdown more specific and more regionally varied than the public narrative had suggested. Mean West and Central African ancestry across the sampled population was between 73 and 82%. European admixture averaged between 19 and 24%. A smaller but consistent Native American signal appeared across the dataset, averaging 1 to 2%, and it was present in a meaningful proportion of individuals rather than concentrated in a few outliers. The geographic pattern within the United States carried its own historical information. African Americans in the Deep South carried higher proportions of African ancestry on average than those in the Upper South or border states. That gradient maps directly onto the differential intensity of slavery across those regions and the differential rates of coerced interracial reproduction that the historical record documents. The DNA was not just showing ancestry, it was showing where the violence had been most concentrated. The most significant finding from the autosomal data was not the European admixture, which the Y-chromosome studies had already implied. It was the African source composition. The public narrative of African American ancestry had centered West Africa, the Senegambia coast, the Gold Coast, the Bight of Benin. What the autosomal data showed was that Central African ancestry, specifically from populations in present-day Angola and the Democratic Republic of Congo, was consistently present at levels the historical and cultural narrative had not reflected. The Bantu-speaking Central African contribution had been demographically significant and historiographically invisible. The genome made it Central African. The picture that emerges from 2 decades of genetic research is clearer than anything the historical record alone could have produced. It is also incomplete in ways that matter. The most significant limitation is the reference panel problem. Genetic ancestry analysis works by comparing a person's DNA to a database of reference populations whose genetic profiles serve as the comparison baseline. The accuracy of any ancestry estimate depends entirely on the quality and coverage of that reference database. And the African continent, which contains more genetic diversity than any other region on Earth, is the most underrepresented region in global genomic databases. Many African ethnic groups, particularly from interior Central Africa, the Great Lakes region, and parts of East Africa, have limited or no representation in the reference panels used by both academic researchers and commercial testing companies. What this means in practice is that some ancestral signals are misassigned. A genetic signature from a poorly represented Central African population may be attributed to a better represented West African one simply because the comparison data for the correct source population does not exist. Ancestry estimates for African Americans are more accurate than they were 20 years ago. They are not as accurate as the precision of the language used to describe them implies. The commercial testing problem compounds this. Consumer DNA kits present their results with a geographic and sometimes ethnic specificity that the underlying science does not fully support. A result that reads 42% Nigerian or 31% Congolese carries an implied precision that population geneticists would not claim in a peer-reviewed paper. For African American consumers specifically, who have the most at stake in recovering what was deliberately destroyed, the gap between what the tests appear to say and what they can actually establish is widest. There is a deeper limitation that no improvement in reference panels will resolve. DNA recovers population history. It traces where genetic lineages came from in aggregate. It cannot tell you who a specific ancestor was, what language they spoke, what they believed, or what they survived. The history it recovers is real. It is also partial. The communities whose ancestors that history belongs to are increasingly the ones deciding how that partiality should be acknowledged and what research would be worth doing to reduce it. The science did not stay in the laboratories. Over the past 2 decades, something shifted in who was asking the questions and who was controlling the answers. In 2003, geneticist Rick Kittles co-founded African Ancestry, a company built on a specific premise that differed from every commercial DNA testing service that preceded it. The reference database was built from African populations, not European ones used as proxies. The stated purpose was not ethnic percentage estimation, it was lineage matching, connecting African American individuals to specific present-day African ethnic communities through maternal or paternal haplogroup identification. The science was the same science. The intention behind it was different. Kittles had grown up in a community where the question of African origin was not academic. He built a tool designed to return an answer to the people the question actually belonged to. The results, for families who pursued them, were sometimes transformative. Not because a percentage on a screen restores what slavery destroyed, but because a specific lineage connection, a maternal line tracing to the Tikar people of Cameroon or a paternal line linking to the Yoruba of southwestern Nigeria, gave something to hold that had not existed before. Some families have made contact with the communities their ancestors came from. Some of those contacts have become sustained relationships between African American families and African communities separated by the trade and reconnected by the genetics. The research also entered territory that extends beyond personal identity. Genetic evidence documenting the scale, the routes, and the demographic consequences of the slave trade has become part of the broader conversation about historical accountability. The DNA does not resolve those political and legal questions, but it changes the evidentiary landscape. It makes the history harder to abstract. There is also an inversion worth noting. The same genetic research that has helped African Americans recover suppressed origins has revealed, in some American families who did not identify as Black, African ancestry that documented history had erased or hidden. The erasure ran in multiple directions. The science is recovering history from all of them. The question of what is done with that recovery, who it belongs to, and what it obligates remains open in ways that no genome can close. For most of American history, the specific origins of African Americans were considered permanently lost. The system that destroyed that knowledge was deliberate, and it was thorough. What the genetics recovered was not everything, but it was more than anyone had reason to expect. Population-level evidence of where people came from, which routes carried them, which erasures were most complete. The history was always there, encoded in the DNA of millions of living people. Science learned recently how to read it. What gets done with that knowledge is still being decided.